All right, so that last video started with me showing you the slide that says DNA, RNA, and protein synthesis, then jumping back to the first set of notes a little bit and talking about just kind of how these different sections were connected and why I was doing what I was doing. Real quickly, again, we've got to know the DNA notes that we started with the first day of school, okay? Then we come to here. We're talking about here that DNA is used to make RNA, and then RNA is going to be used to make proteins. Okay, covered the basic structure of DNA, and then transcription on that last video, right? So as far as transcription goes, we said that that is where, I'm switching papers again, we said that that is where, and sorry, this is kind of a crummy copy of this, but we said that that's where DNA is going to be used to make messenger RNA in the nucleus, because DNA is stuck in the nucleus. It cannot leave. And so proteins have to be made at the ribosome, and so the mRNA is a throwaway copy of the information stored in the DNA. So now we're going to go to this next slide, which is where I had started, that says the genetic code. Okay, so as far as explaining this, here it says genes code for proteins, again, which are going to control the expression of traits. We've talked about that several times this year. We have sections of DNA, and then therefore this would kind of turn into sections of messenger RNA, but sections of DNA that are expressed in our DNA and that are going to code for the traits. Those are called exons because they are expressed. Then we have introns as well. These are segments of DNA that do not code for a trait. So they're going to be cut out of the messenger RNA after transcription, but before proteins are made, right? So on this diagram over here, if this is a segment of DNA, you've got your intron regions shown in blue and your exon regions shown in that kind of yellowy gold color. So the exons are going to be joined together to make the messenger RNA. The introns are going to be cut out of that strand of DNA before the messenger RNA is made, okay? Or sorry, after transcription, but before the proteins are made, okay? And so that being said, then where we're going from here, we're going to be focused on messenger RNA next, okay? So remember, in DNA, adenine always pairs with thymine and guanine always pairs with cytosine. In RNA, there is no thymine, okay? Guanine and cytosine are still going to pair. But then now, instead of thymine, uracil is going to replace that. Okay, and that's going to become important, and you can see some U's for uracil down here. So, what does this genetic code mean? We learned first semester that the monomers of proteins were amino acids. There are 20 of them. We looked at that very briefly in a little activity. We only worked with 4 of them, but we had a chart that had all 20 in them. Well, how does this work, okay? Or how does DNA essentially code for amino acids and then therefore for proteins? So by definition, we've got to know what a codon is. As it says, it's each set of 3 consecutive nucleotides, specifically on mRNA, that are going to code for a specific amino acid. So here you've got 4 examples of codons: AUG, UAC, CCA, and AAG. So that's adenine, uracil, guanine. Okay, hang on. All right, so after transcription, along the length of a strand of messenger RNA, codons are going to be read essentially by the cell or by the ribosome in sets of 3 bases, or the sets of 3 bases are codons, and that's what's going to be read by the ribosome. All right, this is known as the universal genetic code, okay, and what we're looking at here on this particular slide. So we know we have adenine, cytosine, uracil, and guanine as our 4 bases. So the 4 bases combined in sequences of 3 amino acids, in sequences of 3 bases, create 64 possible codons. Those 64 codons are going to code for all 20 amino acids in nature, okay? This is one form of a codon chart, and then I'm going to teach you how to read another form as well. So on this particular chart, you're going to start in the middle, okay? And you're going to work your way out. So let's say I said, I want you to tell me what CUU codes for. Okay, CUU. Right, so CUU codes for leucine. Okay, I'm going to give you another one, and I want you to just do it real quick, and then I'll pause for a second again. Okay, AAC, okay, AAC codes for asparagine. All right, that's what asparagus is named for. We'll talk about that eventually. All right, so 61 of these codons are going to code for amino acids. 3 of these code as stop or termination codons, and that's going to tell the cell to stop making a protein. All right, you can see your stops. You've got UAA, UAG, and then UGA. Those are all of your stops, okay? Each codon is going to code for one specific amino acid, right? But you'll notice that some amino acids are coded for by more than one codon, okay? So for example, UGG codes for tryptophan. That's the only codon that codes for tryptophan. But one of the first ones I gave you, or the first one I gave you, was CUU. Notice that not only does CUU, but also CUC, CUA, and CUG code for leucine, right? Leucine is not anywhere else. Oh, yes, it is. Sorry. It's up here. UUA and UUG also code for leucine. So leucine has 6 codons total that will produce leucine when making amino acid sequences, okay? Then you have some that have 2, okay? Like asparagine only has AAC and AAU. Some of them have 4, okay? Codons that are going to code for them. All right, so you have this codon chart that I attached to your notes, okay? So how do we read this kind? And this is your typical codon chart kind. Historically, this is what came out first. The round one is, to some people, more user-friendly. This particular chart, the only thing you can have written on this little piece of paper is your name. If you have anything else written on it, you can't use it in class if I tell you that you're going to use it on a quiz or something like that, okay, or test. So how do we read this one? Notice it says first base, second base, and then third base going across the top. So you read your first base down the left column, second base across the top, and then third base going down the right, okay? So let's start with, I want you to find AUG. Okay, A, U, then we follow down into the A region, G, okay? So that's going to code for methionine. That's the amino acid that AUG always codes for. Notice it also has the word start next to it, okay? So AUG is going to be the first codon to code for methionine in every single protein that's made in nature. This starts the protein synthesis process. So that means that in DNA, TAC, okay, is what was in the DNA that starts protein synthesis. That then gets transcribed into AUG in the nucleus. The AUG gets read by the ribosome as methionine. So methionine is going to be the first amino acid in every single protein that's made in nature, okay? Let's do, how about CCA? Okay, CA gives you proline. Okay, let's do GAC. GAC, aspartate. Okay, you'll notice that the majority of these end in INE. Okay, some of them do not. Like aspartate is also known as aspartic acid. Right, there's a few that are not going to end in the INE, but most amino acids do. There's your 3 stops up there, UAA, UGA, UAG. This you just need to memorize that AG always codes for methionine and starts protein synthesis. Okay, so how does this work? So after you've got messenger RNA that was made from DNA in the nucleus during transcription, that has to happen first. Messenger RNA is going to leave the nucleus. It's going to go out into the cytoplasm. A ribosome is going to hook onto it, okay, and then it's going to be read or translated using, by you as a human, okay, that amino acid codon chart, or that mRNA codon chart, okay. In our cells, of course, the cell's just going to do this from a chemical and physiological standpoint, okay. So next, our next step is called translation or protein synthesis. So by definition, it's the process of converting the information that's in an mRNA sequence into a sequence of amino acids to build a protein. Right, so this is taking the information in the messenger RNA and translating it into a protein. Those are two different languages. So just think of this as if you were in a foreign language class. When you're translating between Spanish and English, French and English, Latin and English, that you are just moving from one language to another. This is going to happen at the ribosome. Once the mRNA, the ribosome hooks onto the mRNA, then it's going to get, that mRNA is going to be read. Okay, this is going to involve the tRNA anticodons. So they are little interpreter or translator molecules. Okay, so the analogy that I'm going to use for this is, let's just say that I decide to go on a trip and I go to Germany, right? And I don't speak any German, okay? And I meet some people who don't speak any English, right? Let's hope that we have somebody in our little group that actually speaks both languages, that speaks English and German. And so they are going to serve as our translator so that I can communicate as an English-speaking person with somebody that is a German-speaking person. Okay, we need that translator person, or in this case, the tRNA translator molecule. On the tRNA, there's 3 bases that are called an anticodon. Okay, and I'm going to show you something else about that in just a second on the next slide. So there are 3 bases on this loop-shaped structure. The anticodon is complementary to the mRNA codon, right? So adenine in mRNA will pair with uracil in tRNA. Uracil in mRNA will pair with adenine in tRNA, okay? And then guanine and cytosine are always going to pair, okay? Again, no thymine in RNA whatsoever, right? And so then each tRNA anticodon is going to have a specific amino acid that's attached to it. Okay, so on this diagram, this is out of an old textbook that just shows this really simply in nice colors and whatnot. So here again, each tRNA anticodon is going to pair with the proper mRNA codon. This little blobby thing that's blue and kind of tan, that's the ribosome in the background. You can see it labeled here. Okay, so mRNA was made in the nucleus through the transcription process. It's left the nucleus via a nuclear pore. It's out in the cytoplasm. The ribosome hooks onto it. Okay, so this AUG, again, is going to start protein synthesis. It's always going to code for methionine. Okay, notice your base pairing rules. Adenine and uracil always match. Guanine and cytosine match. And messenger RNA versus tRNA, okay, according to complementary base pairing rules. Then we have our next codon. So we have AUG as our first one, then UUC as our second one. So you read your codon chart. AUG codes for methionine, right? UUC, find your codon chart as you work through this, is going to code for phenylalanine, okay? So the tRNA anticodon that always brings amino acids, always brings methionine to the ribosome is going to be UAC. The tRNA that always brings phenylalanine to the ribosome is going to be AAG, okay? If these are your codons, right? And then we have AAA next is a codon. Notice UUU is the tRNA bringing the lysine in, right? So the next slide, all right, so then the next slide, notice that the AUG is no longer in the ribosome, nor is the UAC. So once the base pairs in the mRNA and the tRNA match up, that sends a signal to the cell to drop off the amino acid. That's the green methionine, okay? The ribosome is going to move, and that's going to shift the placement, and I'm not going to get into the terminology of these, the placement of these first 3 codons that we can see here. AUG is no longer in the ribosome. UAC is left as well. Now we've shifted the UUU and the AAA, okay, into different locations, right? So then signal's going to get sent to the cell that these match up. That's going to drop off that phenylalanine, and this chain is going to start to grow, and this tRNA is going to leave as well. And then the AAA will be in this position, and then whatever this is going to come in and be where the AAA is. Okay, and then eventually that lysine will get dropped off. Okay, and we're starting to build a polypeptide chain over here. Okay. So peptide bonds are going to join amino acids together. Okay, so this is known as a peptide bond. We saw this during first semester. Okay, and so because there are many of those peptide bonds to join amino acids, a short sequence of amino acids is called a polypeptide chain. In a true protein, you have multiple polypeptides joined together to make an actual protein. Okay, there's never just one polypeptide. All right, notice what's happening here. GUC is going to code for, I don't have a codon chart in front of me at this moment. GUC, hang on, GUC codes for valine. Okay, so that yellow amino acid is going to be valine. Notice UGA, okay, there's no tRNA coming in. That's a stop codon. So that's telling the cell, stop making this protein. So this will be the last tRNA to leave, and then this polypeptide chain will break off. It'll join with other polypeptides and form a protein in the cells of the body. All right, so then the cell will typically read one mRNA strand multiple times and make multiple copies of the same polypeptide before that mRNA breaks down and before the ribosome breaks apart. These tRNA molecules are going to go back out into the cytoplasm and pick up other amino acids of the same type. Okay, so another methionine in this case, over and over again before they get worn out and break down. The other thing I wanted to mention is that along the length of this loop-shaped structure, there are actually bases along that whole loop. That's your single strand. It just gets organized in this shape, and only these three bases are we concerned with in this particular class. Okay, but that's really a ton of bases along the length of that. If you have any questions, let me know.